Welcome everyone, and thank you for virtually joining us for the latest version of the Variant Effects Seminar Series. My name is Diego, and I'm a member of the VESS Organizing Committee, along with Stephen Airwood and Mireia Soma. These seminars are made possible with support and a lot of help from Laura Muffley and Alex Hopkins, among several others, and our sponsor, the Atlas of Variant Effects Alliance. So a big thank you to them. Also, we're looking at a few more people to the VESS Organizing Committee. So if helping run a seminar series seems interesting to you, then please reach out to one of us. You can find our contact information is listed on the VESS website. As usual, before jumping in, I just have some housekeeping items to mention. Each talk is about 20 or 25 minutes, so there should be time for some questions at the end. Please post those questions, any questions that you have at any time, including during the talk, to the Zoom Q&A. And we'll read these questions out to the speakers at the end. If you have additional questions after the end of the seminar and you're an AVE member, feel free to visit our seminar series Slack channel for more discussion. And finally, we're posting updates on our Twitter account, so be sure to follow us there at Variant Effects. You can catch the seminar today and previous presentations that have been recorded on our affiliate CMAP YouTube channel. Don't worry about trying to copy that whole URL. I'm going to post a little link in the chat shortly. All the details of this seminar series can be found on our website, including a regularly updated schedule of upcoming speakers. I just wanted to briefly mention that we have already scheduled 2 fantastic speakers for the September seminar, so be sure to add that to your calendar now. And for the last housekeeping item, this hasn't really been an issue, but as a reminder, please be considerate when posting comments or questions. We expect everyone to be respectful and use welcoming and inclusive language. Without further ado, we have 2 great speakers scheduled for today's presentations. Starting us off, Sarah Gersing is a PhD student in Rasmus Hartmann Peterson's group at the University of Copenhagen. Sarah's work focuses on characterizing the effects and mechanisms of variants in the human enzyme glucokinase using deep mutational scanning in yeast. The title of her talk is A Comprehensive Map of Human Glucokinase Variant Activity. Sarah, feel free to start sharing your screen and go ahead and start whenever you're ready.
Thank you for the introduction, and thank you to all the organizers for inviting me to talk today. As mentioned, my name is Sarah Gersing, and I'm a PhD student at the Hartmann Peterson group at University of Copenhagen. And today I'll talk to you about this really cool enzyme, glucokinase, and how we use yeast to generate a comprehensive map of human glucokinase variant activity to aid in the diagnosis of monogenic diabetes, and also to gain a better mechanistic understanding of variants in this enzyme, which may inform the development of drugs targeting glucokinase to treat type 2 diabetes. So glucokinase is known as the body's glucose sensor, as it regulates insulin secretion in pancreatic beta cells. And glucokinase can do this by regulating or by catalyzing the rate-limiting step in this pathway, which is the phosphorylation of glucose to form glucose 6-phosphate. What allows glucokinase to play this sensor role is its kinetics. Compared to the 3 other human hexokinases, glucokinase has quite a low affinity towards glucose, around 7.5 to 10 millimolar, which is comparable to plasma glucose levels. In addition, glucokinase shows sigmoidal kinetics, which allows the enzyme to respond rapidly to changes in plasma glucose levels in the physiological range. These sigmoidal kinetics are a bit unusual, as glucokinase functions as a monomer and only has 1 active site. Instead, the sigmoidal kinetics arise due to conformational dynamics. So simply speaking, glucokinase exists in 3 different conformations: the closed active conformation, the open conformation, and the super open inactive conformation. At low glucose levels, glucokinase predominantly exists in the super open conformation, and during catalysis shifts between the super open, open, and closed conformation, which results in a slow catalytic cycle. At higher glucose levels, glucokinase bypasses the super open state and binds a new molecule of glucose before the slow transition into the super open state can occur. And this results in a faster catalytic cycle. The ratio between these two cycles, which depends on glucose levels, give rise to the sigmoidal kinetics that are essential for proper glucokinase function. So due to this important role of glucokinase in glucose homeostasis, variants that alter activity are associated with disease. Heterozygous variants that increase activity are associated with hyperinsulinemic hypoglycemia, while variants that, or heterozygous variants that decrease activity are associated with hyperglycemia and a mild form of monogenic diabetes known as glucokinase modi. So glucokinase modi is estimated to affect around 10 million people worldwide. However, these patients often do not require treatment. They are, however, unfortunately frequently misdiagnosed as having type 1 or type 2 diabetes and therefore receive unnecessary treatment. This can be prevented by correct diagnosis, which is obtained by genetic testing and variant interpretation, but this is complicated by the effect of many variants remaining unknown, as is of course the case for many teams. So the aim of this project was to generate a map of glucokinase variant activity, first of all due to the clinical relevance, as mentioned, especially for glucokinase modi patients, but also to gain a better mechanistic understanding of glucokinase variants, as glucokinase is the target of drugs known as glucokinase activators that are currently being developed to treat type 2 diabetes. But first, to generate a map of variant activity, we needed an assay to assess the activity of glucokinase variants. And for this, we used yeast. So we generated a yeast strain deleted for the 3 hexokinase genes that shows severely reduced growth on glucose medium. However, when we express wild-type human glucokinase, this rescues growth at various glucose concentrations. We also tested 8 variants and their ability to rescue the growth defect, including 2 common variants assumed to be benign, a catalytically dead variant, and 5 pathogenic variants. And as you can see, the 2 common variants show growth and therefore activity comparable to wild type, while the catalytically dead variant show decreased growth, as does 4 pathogenic variants associated with decreased activity and glucokinase modi, while 1 pathogenic variant known to be hyperactive and associated with hyperinsulinemic hypoglycemia showed increased growth compared to wild type, showing that this assay is able to detect pathogenic variants, including both loss and gain of function. Glucokinase activity, of course, also depends on the level of its substrate, glucose. So we also tested complementation at various glucose concentrations and found that a concentration of 0.2% glucose enabled the detection of both the loss and gain of function variants. In addition, this concentration corresponds to 11.1 millimolar, which is quite close to the affinity of glucokinase for glucose. So having established an assay to detect glucokinase activity, we next multiplex this assay based on protocols from the Roth lab. So we expressed a library of glucokinase variants in the yeast triple deletion strain and grew the yeast cells on medium with 0.2% glucose to select for glucokinase activity. Then we sequenced variants before and after selection in tiles of around 150 base pairs And following sequencing and analysis, we determined the relative frequency of each variant before and after selection to obtain variant activity scores. And we obtained variant activity scores for 97% of the possible variants. And we were able to score both variants with decreased activity shown in red, as well as variants with increased activity shown in blue. And we found that around 50% of the variants had decreased activity, while around 14% of the variants had increased activity. When we map the median activity score at each position onto the structure of glucokinase, as can be seen here with glucose shown in green, we found that surface exposed positions were generally resistant towards mutations, while active site residues and other buried positions, here variants tended to decrease activity as would be expected. In contrast, positions where variants increased activity shown in blue concentrated at a previously known allosteric activator site. So these observations provided an initial validation that the yeast assay or the activity scores obtained in yeast reflected human glucokinase activity. But to further validate the score and also to assess what aspect of activity that the assay reflects, we examined the correlation of activity scores with different kinetic parameters of 38 variants previously examined in vitro. And we found that the activity scores correlated quite well with the catalytic efficiency of the glucokinase variants, such that an increased activity score was associated with an increased catalytic efficiency. And this held true for both variants with decreased activity as well as variants with increased activity. So having examined the relationship of the activity scores to in vitro glucokinase activity, we next wanted to examine how they correlated with fasting glucose levels in carriers of glucokinase variants. So here we found that a decrease in activity score was associated with an increased level of fasting glucose, as would be expected. However, glucokinase is of course not the only factor that influences fasting glucose levels in carriers of variants. There are also other genetic and environmental factors that affect fasting glucose. So this suggests that our assay is at least somewhat able to capture fasting glucose in carriers of glucokinase variants. These validations were, however, limited by the number of variants that had previously been examined. So to more broadly validate our results, we examined glucokinase evolutionary conservation across species. And we included both gluco and hexokinases in the multiple sequence alignment that is used as input for the analysis. And the output of the analysis is an evolutionary distance score, which quantifies the likelihood of a given substitution. A score of 0 corresponds to a variant being compatible with function, while a high negative score suggests that a variant is detrimental. And here we then see the positional median score for evolutionary analysis and activity compared. And we can see that they agree quite well on regions that are tolerant towards mutations shown in light colors, as well as regions where variants tend to decrease activity. However, the evolutionary analysis did not seem to capture regions where variants increased activity, as well as the mutational sensitivity of the region spanning from around residue 150 to 200. The mutational sensitivity of this region, as well as variants with increased activity, likely arise due to the conformational dynamics and sigmoidal kinetics that are characteristic for glucokinase compared to the other hexokinases. And therefore, these effects are unlikely to be captured by the evolutionary analysis, that included hexokinases more broadly in the multiple sequence alignment. But except for these regions, the evolutionary analysis generally supported the activity scores, especially for regions that where variants decreased activity, as well as those that are tolerant. So having generated a map of human glucokinase activity and having validated the map, we next wanted to examine the clinical relevance of the activity scores. To do this, we used 68 pathogenic variants previously characterized, or that had previously been functionally characterized. 60 variants were associated with glucokinase modi, while 8 variants were associated with hyperinsulinic hypoglycemia, and we also had 3 benign variants. We didn't have a score for one of the benign variants and therefore used an imputed value, as we were already quite limited by the number of benign variants. So looking at the distributions of the activity scores for these 3 groups of variants, we can see that variants associated with glucokinase modi are generally associated with decreased activity, as would be expected, while variants associated with hyperinsulinemic hypoglycemia show increased activity, and benign variants are associated with a wild-type-like score. To further examine the clinical relevance, we also looked at the nomad allele frequency and activity scores of glucokinase variants found in nomad. And as you can see, the more common variants tend towards a wild-type-like score of 1, while the rarer the variants are, the higher the span in activity score, including both variants with decreased activity and increased activity that might be pathogenic. So having examined the clinical relevance of the glucokinase activity scores, we also wanted to use the activity assay to gain a better mechanistic understanding of glucokinase variants. And we speculated that some variants might affect the stability of the glucokinase conformations differently, and thereby shifting glucokinase towards either the inactive or active conformation to decrease or increase activity. So to examine this, we used Rosetta to predict the changes in protein stability for both the super open inactive conformation as well as the closed active conformation. And then we calculated the difference between these two to obtain a delta G score. For the delta G, white regions corresponds to positions where variants are predicted to affect the 2 structures equally, while red positions are where variants tend to destabilize the closed active state compared to the super open, while blue positions are where variants are predicted to destabilize the super open compared to the closed. When mapping these scores onto the structures of glucokinases, we can see that red positions that are predicted to destabilize the closed conformation are found at buried positions in that state, but are more surface exposed here in the super open conformation. And therefore, variants at these positions might shift glucokinase towards the super open inactive state by destabilizing the closed conformation. In contrast, blue positions where variants are predicted to destabilize the super open conformation are concentrated at the allosteric activator site, and therefore hyperactive variants at this site might increase activity by shifting glucokinase towards the closed active conformation by destabilizing the super open state. Previously, 6 hyperactive variants in glucokinase had been shown to increase activity by promoting increased populations of the closed state, but by combining our activity scores with predictions of protein stability, we identified 467 hyperactive variants that might increase activity through this mechanism. So to summarize, we used yeast complementation to generate a map of human glucokinase variant activity that included both loss and gain of function variants. The activity scores could distinguish pathogenic variants from benign, and that is especially important for patients with glucokinase modi, where correct diagnosis can lead to or can prevent unnecessary treatment. We also use the activity scores to gain a better mechanistic understanding of especially hyperactive variants found in the allosteric activator site. This site is the target of drugs known as glucokinase activators that bind the site and mimic hyperactive variants and are being pursued to treat type 2 diabetes. And therefore an increased understanding of hyperactive variants in this site and their mechanisms might inform the development of these drugs. Finally, I would like to thank all the people involved in this project, especially Matteo, who did the majority of the computational analysis in this project, Mayanella, who generated the yeast deletion strain together with Gerish, and also did some initial complementation tests, and Atina, who provided me with protocols and guidance on the MAVE experiments. And with that, I'm happy to take any questions.
Thanks a lot, Sarah. And please, everyone, remember that you can post your questions in the Q&A box below. And while we wait for some questions from the audience, maybe I can start with one question. And I was wondering if you have an hypothesis for those mutations that destabilize both structures, the closed conformation and the open conformation. Like, do you have an hypothesis on what's going on in there? Are they just destabilizing some other part of the protein?
Yeah, if they affect both structures, what happens with those mutants? Is that what you're asking? Yeah, I think that's just if they're just destabilizing the protein, then that would just lead to low abundance and disease through that mechanism. An important part of this analysis is also that the variants that might act through this mechanism also need to not overly destabilize either of the conformations, because then that would just lead to degradation of the protein.
And do you have any control in your experiment for protein expression, for example? Like, because you would not distinguish something that doesn't grow because it's just right?
Yeah. So our activity scores also includes, of course, variants that are of low stability, for example. So that's more like a functional score that includes many types of mechanisms, of course.
Okay, thank you. And I have another question here, and it's a bit more in terms of general rules or guidelines when you think about the MAVE assays. Why do you prioritize to use yeast as a model system versus something else like a cancer cell line that would mimic maybe better the phenotype in patients? Is just in terms of numbers, or like the experiment becomes easier, for example?
Yeah, I think definitely using yeast makes experiments quite easy compared to others. But I think it's also important to say that at least for glucokinase, the phenotypes in yeast appears to quite closely reflect also what you see in humans and in patients.
Okay, great. Thanks a lot, Sarah. So if we don't have any more questions, anyway, remember that you can still post your questions if you have questions for Sarah in the chat.